Hello, welcome to a look at passive cell transport. So when we're looking at the cell membrane, there's a couple of things we want to look at. So here we have the phospholipid bilayer, which is what all of the things pass through. Next, we have the transport proteins, also known as ion channels, which help some other things pass through it, like if in facilitated diffusion. Next, we would have these like carbohydrates that are sticking out. Those are kind of used as like identification tabs. Tags, sorry, not tabs. So they're kind of used as identification tabs so that other parts of your body know that, hey, this has this carbohydrate. So it's like our cell, it's one of us. All right, and this is just another look at it from sideways with everything labeled. It's known as a fluid mosaic model because some people describe it as a mosaic because you can kind of see the similarities here. It looks like that, like kind of like wavy with a bunch of stuff in between, and you can compare it to the mosaic. A bunch more diagrams here. So, like I said earlier, the cell membrane regulates what enters and leaves the cell, and this is called the phospholipid bilayer. So, the structure. There's two layers of lipids, hence why it's called bilayer. The proteins forms gates, channels, or pumps that move materials inside and outside the cell, and carbohydrate chains are cell identification, so cells can identify each other. All right, transport. Permeable means that the membrane allows all substances to pass through, such as when you're putting like water through a strainer, all the water will fall through. Selectively permeable or semi permeable, only certain molecules will be allowed to pass through. So, certain things cannot pass through if they're too big or like strongly charged. So, for example, spaghetti cannot pass through that a strainer because it's too large, it won't fit in the holes. A screen on a window, fresh air can pass through, but the bugs cannot. You can look at your window and see most bugs cannot pass through. However, fresh air obviously can. Right, equilibrium in a cell. So the particles pass through the membrane without any added energy in order to reach equilibrium. So equilibrium is basically whenever there's, let's say, like three things here and three things here. This would be an equilibrium state. So basically, what would happen is these two would constantly shift back and forth in between each other, and there would be no net change in concentration on both sides. All right. Next slide. So this is just kind of like a vocabulary key for the next parts. Solute is the substance being dissolved. Solvent is the liquid that the solute is dissolving in, and the solution is a mixture of a solvent and a solute. So an example of this would be, let's say, a cup, and there's a bunch of water in the cup. So the water would be the solvent. Now, if I take a salt shaker and I pour a bunch of salt into the water, the salt would be the solute. So you can kind of remember that as like solute, salt, and then the solution would be the mixture of the two, which would be salt water. All right. So concentration gradient in passive transport, which is what we're looking at today. Everything moves from a high concentration gradient to a low concentration gradient. So let's say there's like five things here. There's like two things here. Well, this wants to reach the equilibrium state, so things will move from this to this side until it reaches equilibrium. All right, so there's two primary ways that molecules can pass through membranes, which is what we're looking at, passive transport. So the molecules will pass through the membrane from high to low without the use of energy, so this will not require any energy for this to move from here to here, because moving from high to low. Now, sometimes our body wants stuff in a certain area. So, like, let's say there's two things here and there's three things here, but our body needs some of this stuff over here. So then it would be active transport. It would move from here to here, which would require energy. You can kind of think of it as like a boulder rolling up or down a hill. So for passive transport, you would roll the boulder down the hill from the high point to the low point, which doesn't require any energy. Gravity will do all the work for you. However, for the other situation, when you're rolling it uphill, you have to put in a lot of work to push the boulder uphill. Another example you could think of is perfume. Whenever you spray perfume in a room, it automatically spreads out throughout the room to reach equilibrium. It'll go to the areas without perfume. However, if you want to like cluster the perfume in another area, you'll have to like push winds and like try and like force the perfume to be in the area. It requires energy, obviously. All right. Three ways of passive transport across the cell membrane. So first is diffusion, which is just movement of molecules. Next, facilitated diffusion. So movement of molecules with help from transport proteins. And osmosis is just the diffusion of water. It is just the diffusion of water. All right, diffusion. It's where molecules move from a high concentration gradient to a low concentration gradient through a membrane. Examples would be food coloring in a beaker or perfume in a corner of the room. They'll all like spread out. The food coloring will color the water, and the perfume will spread out throughout the room. All right. Passive transport through the lipid bilayer. The molecule would go here, would go through the membrane, go on the other side. Diffusion. So a bunch of dye would be here. The dye would go like this until it reaches equilibrium on both sides. Here's another example. So there's two solutes here. So there's like a bunch of this on one side, a bunch of this on one side. Some of this stuff will move here, some of this stuff will move here until there's about an equal number on both sides. Next, facilitated diffusion. So facilitated diffusion, like I said earlier, is whenever something needs help to diffuse, like glucose is too large to go through this. Like it's like, let's say it's like this big. Well, it can't go through that. So instead, it'll go through here where it'll be like pushed with like pumps and stuff through the transport protein and end up on the other side. Here's like some examples. Like this is kind of like a cool thing. So it's like a corridor. So something will go here, then it'll spin around and it can leave through here and nothing can enter. So it's kind of like those like cool doors you sometimes see. Right. Facilitated diffusion when a cell membrane protein facilitates or helps a molecule diffuse through the membrane. This is how it looks like in passive transport. All right. That's how the other thing looks like. Type of facilitated diffusion. This is an ion channel, so it's basically a transport protein with a polar pore, which ions can pass through. So ions are basically things that are like that have like a charge different from the regular charge. So you can learn more about this in chemistry, but you don't have to worry about what types of ions there are for now. Instead, just know ions pass through an ion channel, which is a special type of transport protein. Finally, osmosis, the diffusion of water through a selectively permeable membrane. So cell membranes are completely permeable through water, meaning water can always pass through them without having a dramatic effect on the cell. So isotonic is when the concentration on each side of the membrane is the same. So let's say you have a cell and outside you have water. So let's say this is 50% water and this is also 50% water. So this would be isotonic. Like I mentioned earlier, stuff would be moving in, stuff would be moving out. It would just be in equilibrium. Hypertonic, above strain. Solution has a higher solute concentration than the cell. So in this case, I'll actually need a bigger thing here. I'll just draw it over here. All right. So large cell. All right. Let's say this has 60% water. Well, then we would know it has 40% solute. So that means I will know it doesn't mean, but like let's say this has 70% solute and 30% water. Well, we would know that this is a hypertonic solution because it has a higher solute concentration. And then in this case, osmosis would occur and water would move from the area with high concentration. So this would have a higher concentration of water, not solute. Water would leave the cell, making the cell shrivel up. That's why you don't want, like, that's why you like get shriveled if you stay in water for too long. And it's also why you don't put a saltwater fish in freshwater because freshwater would be hypertonic, meaning water would leave the cells of the saltwater fish. All right, next, hypotonic, below strain. So the solute concentration is lower. Again, I'll have to do it bigger to show the solute concentrations. So let's use the same exact cell. 60% water, 40% solute. In this case, let's say this one has 70% water and 30% solute. So in this case, solute is lower, meaning it's hypotonic, and water is higher, so water would enter the cell. Alright, so these are some example solutions. This is hypotonic, isotonic, and hypertonic. I already explained why. Lower solute, same solute, and higher solute. Remember, this is solute, not water. You can look at it either way, but I find solute easier to explain. Alright, everyday examples of solutions. Hypertonic is alcohol, ocean water, Epsom, salt bath. Hypotonic is distilled water, and isotonic is blood. So, hypotonic solution and hypertonic solution. So the thing, water is going from the hypotonic to the hypertonic solution through reductively permeable membrane. Here you can see the increase in water on this side. All right. So this is what happens to animal and plant cells under each type of uh, solution. So what I want you to take note of is when the cells are normal. So for animal cells, this would be in isotonic, and for plant cells, it is when they are turgid, which only occurs when they are hypotonic. However, if animal cells are hypotonic, then they become lysed and they get too much water in them, so they blow up. And whenever they're hypertonic, they shrivel up because water is leaving them. However, plants don't experience this whenever they are in a hypotonic solution. The reason why is because they have a cell wall while we don't. So the cell wall is kind of like acts as like a strong barrier, so the water inside the plant cell just kind of like pushes out, making the turgid cell look thicker. So it's flaccid in this one. Whenever it's isotonic, you can kind of see it's like more rigid. And here, since all the water is leaving, it's plasmolized. We call the pressure that emits out of the plant inside hypotonic because of all the water, turgor pressure. Alright, so this is just an example of a plasmolized plant cell. And some more general terms. Osmotic pressure, the water pressure in cells. Turgor pressure, the water pressure in plant cells. Plasmolysis, process where the cell membrane pulls away from the cell wall due to water loss. Alright, and that is it for passive transport. Hopefully you do good on your quiz, test, or retest. Good luck!